been all along, confessing twilight on and on, without a clue, without a sign, without grasping it. The real question to be asked: where have I been? I'm a sharpshooter at post masquerade, hiding both face and mind, all free for you to draw. I'm a sharpshooter. What else should I be? Please don't take off my mask, revealing dark. Moments of calm, nothing left to be found. A mirror right in front of me, that's where I found an empty glass, reflecting that's a truth. It's telling words not to be told. I need that mask. I'm a sharpshooter at post masquerade, hiding both face and mind, all free for you to draw. I'm a sharpshooter, chained down to my core. Please don't take off my mask, my place to hide. I can't tell you how to see me. Just a cage of bones, there's nothing inside. Will it always be burning down the walls? Is there a way for me to break? I'm a sharpshooter at post masquerade, hiding both face and mind, all free for you to draw. I'm a sharpshooter, have no face to show. Please don't take off my mask, my disguise.
Hi, everybody. Welcome back for the last time to Chem 131. What do we got going on here? Well, it's just whatever's left, really. I mean, there's only so many more slides. We're going to talk about enzymes today. And then we're going to talk about DNA really briefly. But before we do that, there is a quiz that was due yesterday. It's still open for one more day. So tonight's the last call for quiz 6. There will be another quiz that goes online for this protein chapter and the DNA chapter. And then after that, we have like final administrivia to talk about, like literally the final. So we'll talk about when we're going to put exam 2. I want to move exam 2 to Monday to give people the weekend to study, obviously. But what that does mean is that the quiz for this unit will be very short in terms of time frame. That's probably okay because there's not a ton of stuff that you have to do in the proteins chapter. I want to have it available for you so you can look at the answer key and have that available so you can study for the exam number 2 on Monday. And then we'll have a review session next week on one of the class days, probably Tuesday. And then your final will be Wednesday or Thursday. The final is just like the exams. I schedule it all day. That doesn't mean it takes all day. It just means that sometime during that during that entire day, please take the final. You get 2 attempts like always. The thing about the final is it's long, and you don't get much additional time, and it's just the entire course content randomly assorted. So we'll talk about that at the end. Let's talk about enzymes right now. So what the heck is an enzyme? Enzymes are really important. So what they are catalysts for reactions that would normally go really, really slow. The reactions that are important for life and biology and I can't even describe what reactions these are because it's just all of them, really. Like moving electrons around is slow. Creating energy, not creating energy, but creating the bonds in molecules and then breaking them to get that energy back to do stuff is slow. And if we didn't have catalysts to speed these reactions up to go at a speed that we could sustain, life wouldn't exist because the reactions are just too high in energy. It takes too much energy to make them go. But what enzymes do is they lower the activation energy of a reaction by providing an alternate pathway. For instance, most molecules will react by crashing into each other at high velocity. But an enzyme will hold things together and then just sort of jam them together. So you could think of it kind of like an assembly line that connects parts, where the parts are molecules. And that helps facilitate that because we don't have to wait for 2 molecules to randomly collide with each other. The enzyme just does all that work for us. Now, because it's a catalyst, it's not consumed in the process. So the enzyme can be reused over and over and over again, provided that we are using it correctly. We're going to talk about some conditions in a second that destroy the catalyst. Sorry, I had to sneeze there. When I say destroy the catalyst, what I mean is like we bind to it irreversibly and then the catalyst is no longer useful. Now, as far as I can tell, every enzyme has a common name in biology, and those common names end in ase. And they generally are named based on what they do. So lipase, for example, will hydrolyze lipids. It puts water right up next to the lipid and breaks it, whether there is an acid catalyst there or not. Protease will hydrolyze proteins. Decarboxylase removes CO groups. Reductase will reduce a molecule, and so on and so forth. So there are tons of enzymes that do tons of different tasks. And when you get into biology, one of the main things that you're going to be doing is you're going to be learning what all these enzymes are and what they do. So cell bio is just full of that. For example, I remember when I took a bio class in college, it was the only bio class I took. We had to memorize the Krebs cycle, and they basically gave us all these reactions and all these enzymes, and there's like 16 of them. And I decided that I did not want to be a biologist because I wanted to learn content rather than memorize enzyme names. But so I'm not going to make you do that here because I think that that's trivial. You can always look that information up. I care much more about like what's specifically going on at like the nitty-gritty level of detail. So like what bonds are being formed. Now, what does an enzyme look like? So this shape right here is actually a protein. And what the protein is doing is it is shaped in such a way that allows our molecule or molecules, in this case we're going to join 2 things together. They can sneak right into what is known as the active site. So this region right here that is in blue, that I have drawn and looks kind of like a butt, this is the active site. And what the active site is just the region that the chemistry occurs at. It is shaped specifically for this purpose so that only the molecules we want can fit into the active site, or at least only as few molecules that we want can fit in the active site. So on the right side of each half here is the actual protein diagram, and it's in low resolution because it's just showing the topography of all the R groups. You don't really get to see the protein chain here, unfortunately. And then on the left is a really simplified cartoon drawing where we just sort of have this blob, and the blob has open regions that can encapsulate the molecules that we're trying to react here. Now, an enzyme can have multiple active sites. Specifically, the one that is useful for a reaction is what we call the active site. There may be other places in the molecule, like down here it looks like it's kind of shaped in a particular way. A molecule might go into that space and do nothing. Or, as we might find out, that might change the parameters of the reaction at the active site. So, other parts of the topography of the enzyme are important too. And we'll discuss that in a little bit more detail as we go on. So what are enzymes made of? So the first thing that they're made of is proteins. Proteins are a really common component, but there are also other components to them. So one of those components is called a cofactor. So if you've taken a biology class, you may have heard of acetyl coenzyme A. And what coenzyme A is just basically a vitamin with some other molecules attached to it. I have not taken that many bio classes. I know that acetyl-CoA is used in a bunch of reactions, so we use it to... Oh, goodness, what do we use it for? I think we use it in, like, oxidative phosphorylation. Gosh. I'm not a biologist, so I just tell you the molecules and what's important in them. Now, there are, it's made in glycolysis. Okay, thank you. Now, other things that you can put into your molecule do not have to be organic compounds. You can actually have things that are heavy metals. So for one example of that is the iron in hemoglobin. And if we look beyond human biology, we can actually find other heavy metals in different creatures. So for example, in crabs have copper in their enzymes for oxygen transport, I believe. So it's not just iron that's universal. Depending on how evolution has randomly just come up with these, I don't want to call them features, but I guess that's the best, like, features of various organisms. It's essentially random, and whatever was available or abundant in the habitat of those creatures generally was incorporated into their biology. And so iron is very abundant, and so that's why iron is incorporated into hemoglobin, most likely. And then over billions of years of just reactions coming together and then life being formed, that's how we ended up with these enzymes. So there's nothing inherently wrong with having heavy metals in there. It's just that a lot of them are toxic, but some of them are essential. And all of these can be found as parts of enzymes. Now, when an enzyme reaction occurs, the stuff that you are putting into the enzyme is called the substrate. So the substrate interacts with the enzyme. Oops, I thought the picture was showing up. It's on the next slide. The substrate comes in, and then the enzyme does the reaction. It does either an assembly or a disassembly, depending on what you're trying to do for the reaction. And then it releases the products. And so that looks kind of like this. So in this reaction here, the substrate is a reactant that has two parts to it. There's the rounded part and the triangular part. So these are going to enter the active site, which is already shaped in such a way to accommodate these things. And then the enzyme will just kind of manipulate the substance in some way to facilitate the reaction. So for this reaction, we would be cleaving a bond, whatever bond is here, to make these two products independent. and then those products would pop out. So ignore this right here, because this is the lock and key model, so we're going to assume that it doesn't really change much shape. In the next model, which is more accurate, we are going to talk about the shape of the enzyme changer. But this model is called the lock and key model, and the reason it's called that is because only specific keys will fit into the lock of the enzyme. Kind of like how with your apartment key or your house key, if you try any other key, like your car key or maybe your work key or something like that. I do that all the time. I would try and use my work key to get into my apartment, or my house key to get into the classroom, just because I'm usually very tired when I'm leaving work or coming home or whatever. And obviously it doesn't work, right? So like sometimes the key will fit into the lock, but it won't turn because the tumblers aren't aligned in the correct position. And then sometimes the key is just the wrong shape; it won't fit at all. So our enzymes having these particular shapes force other molecules to just go away. So only the reaction that we are trying to do gets performed. At least that's the theory. Now, as it turns out, and I have actually a really funny story about this, there are only so many keys out there, right? And in the same vein of that, there are only so many shapes. Lots of molecules have similar shapes. They're not exactly the same, but they're close enough, right? My best friend moved into an apartment here in Everett because he got a job teaching at a different college. I think he was teaching at Edmonds. And when he moved into that apartment, we gave each other keys because we were living within 5 minutes of each other for the first time in years. So it's like, oh, okay, you can come over whenever you want to, or I'll watch over your cats while you're away or whatever. So he gave me his apartment key. Well, one night I was really, really, really tired coming back from his place. I think it was like 3 or 4 a.m. We might have been playing Dungeons & Dragons or Gloomhaven or something. I was really tired and I came home. I used his key on my apartment door, and my door opened. And it was because his key was similar enough to mine, because there's only so many key patterns out there, that I learned that, oh, holy shit, his key works on my door. Not only that, it works better because his key was newer. And mine was old and like had started to wear away. My key did not work on his door, but it did fit, and I could lock the door. I couldn't unlock the door. So there are similar molecules out there, just like in this scenario with keys. Like, your apartment key will fit somebody else's lock, just like some other molecule will fit the enzyme. And so enzymes are not perfect. So we can have issues crop up, and we're going to talk about that a little bit later. Now, this model is not really the complete picture of what's going on, because enzymes are not these rigid balloons. What ends up happening instead is when molecules enter the active site changes shape, and so does the rest of the substrate. And so we're going to look at that in this picture right here. So here you can see that the shape of this molecule is obviously not the same as these shapes here of the substrates coming in. This is obviously a exaggeration of what these molecules look like, because these just basically look like puzzle pieces. But when the substrates enter the enzyme, what actually happens is that the shape of the molecule deforms. Not just in the active site, but in other places too. And in doing that, that allows the enzyme to do the reaction. Now, it's important to note that other sites can change too. We just particularly focus on the active site. But later on, what we're going to talk about when we talk about inhibitive I can't think of the word right now. Molecules that inhibit the reaction. Non-competitive inhibitors. That's what I was thinking of. When we talk about non-competitive inhibitors, they bind to other places that aren't the active site, but they change the overall shape, which ends up screwing up the active site enough anyway. So the rest of the enzyme's surface is just as important as the active site for this model, which is called the Induced-Fit Model. So the induced fit model is basically, oh, my active site changes shape to accommodate the incoming molecules, but is still a close enough shape that it will exclude other components that don't belong. So this model is actually called this model is actually the current model of how we believe enzymes work, at least through testing. And we have a lot of evidence to support this that we're going to show you in how do we make enzymes speed up? How do we slow them down? Like, what's effective but what isn't? And it turns out the things that are effective are things that do not denature proteins. So, like, putting it in a high or low pH would ruin the enzyme. And we're going to see that. We're going to look at some data. Now, depending on the enzyme that you're using, some enzymes are much more specific than others just based on their shape and the size of the molecules that fit in them. For example, urease is very particular. Only a specifically shaped molecule will fit in there, and that molecule is urea. So urease will decompose urea into ammonia and CO2. Nothing else will fit, and it particularly works well because urea is such a tiny molecule. So if you have a teeny tiny puzzle slot, you can only put tiny pieces in there. But if you have a giant slot, you could put any piece in that you'd want to that fits that. So, like, for example, you know when you're a kid and you have those boxes where you have, like, oh, it goes in the square hole. You may have seen that meme, right? And so only this shape would fit, but the rest of the shapes, like maybe this giant-ass moon that I'm going to draw, too big, or this huge circle, that's not going to fit. But if your space that you can fit this in is just like this, all of these pieces will go in there. So if you're catalyzing a reaction that has larger molecules, you could put teeny tiny molecules in there or big molecules in there, and it's far less specific. So an example of that is the enzyme carboxypeptidase. What that is it just chops off the carboxylic acid part of your protein, regardless of how far along the protein or how big the protein is or anything like that. It just grabs the end of it and goes hop, and we get rid of that. All right, so let's talk a little bit more about how we can affect enzymes. So the ability of an enzyme to perform its task is called its activity. So an enzyme with high activity means that the reaction is proceeding at a fast rate. So maybe 20 reactions a second or 1,000 reactions a minute. Or I actually don't know the rate at which enzymes work at. I would be surprised if it was much slower than X number of reactions per second. You just do because molecules move pretty quick, right? Now, we can command the activity of an enzyme by manipulating external factors. So the first thing that we want to consider is how much stuff do we have? What I'm going to do is I'm going to draw a bunch of enzyme active sites. So here's an enzyme, here's an enzyme, here's an enzyme, and here, oops, that's a square root. I don't know where that came from. And here's an enzyme. And so the reactions that I want to put in here involve this substrate. So if I have enough substrate to fill all the spaces, then those substances will react efficiently. But if I have anything extra has to wait its turn, or may even just kind of go straight on by unreacted if there are no active sites. If I increase the concentration so that there's tons of stuff in here, but I don't change the amount of enzyme, then all of this is just going to go straight on past. So in regards to how well an enzyme works, obviously you need substrate. But increasing the amount of substrate, so this would be less substrate and this would be more substrate, is only so effective because at some point our enzymes get saturated. And if all these active sites are full, you hit a limit on how many reactions you can do. If you've ever played a factory building game, so Factorio or Satisfactory, or any other game that has the word factory as a pun in its title, probably a factory game, what you end up seeing in those types of games is that if you make products that then go on to the next thing, and then go on to the next thing, and then go on to the next thing, some part of that reaction is the slowest part, and then everything else backs up from there. And you are capped at the maximum speed of making all of your parts in your factory is limited by the slowest step. Well, if this step is completely saturated right here, because I'm out of spaces to put my substrate, this is going to govern how fast my reaction goes. And so the maximum speed that I hit will cap out. Now, how do you fix that? Well, what you do is you introduce more enzyme. As you increase the amount of enzyme, the speed of the reaction increases linearly. Now, this effect will top off too. You will hit a maximum if you just don't have any substrate available. But in biological media, the most common limit is the enzyme. There's generally plenty of substrate to go around, and in fact, there can be too much substrate, which can lead to interesting conditions or possibly bad conditions. So those are two ways that we can control the activity of an enzyme. The first one, changing the concentration of substrate, is only so effective. At some point, we tap out on the benefit that we get from that. But the concentration of enzyme has no such limit, at least in practical conditions. All right, what else can we do to enzymes? Well, the things that are available to us are the same things that denature protein. So temperature and pH are things that we can manipulate, but we have to do it carefully. For example, with temperature, if you heat up a chemical reaction, the reaction will go faster and faster and faster the higher the temperature it is. But with enzymes, that's not the case, because in this part of our temperature range, we have, see, the reaction is increasing in its rate because reactions do go faster at higher temperatures. So that is true even in biology. But what ends up happening is we don't end up just going up and up forever. At some point, the activity decreases because the protein denatures. And specifically, that's the enzyme. So same reason that if you heat up egg white or meat or whatever, it changes color because it's denaturing. The same thing happens if you increase the temperature of your protein. The activity goes down pretty quickly. So there is some optimal temperature at which a reaction will occur. And that would be the peak of this graph. So as temperature increases, we can make the reaction go faster, but only up until a point. Now, the same thing that's true of temperature is true of pH. There is some ideal operating pH at which an enzyme will react. And so if I plot a graph of enzyme activity versus pH, there's going to be some optimal pH at which the enzyme undergoes peak efficiency. But if I stray too far from that, then either I would denature my protein, or maybe I'm reacting with my compounds, or I have the wrong compounds present. The range and also the optimum pH depends on the enzyme that you're looking at. Some enzymes operate at acidic conditions only. Others tend to work only around physiological pH, which is like 7.35. Generally speaking, enzymes are pretty sensitive to pH changes. If you change by like a pH unit of 1 or so in either direction, you tend to see this activity fall off pretty quickly. This is particularly true in the body. If you have the pH of your blood goes below 6.8, it's normally about 7.3. But if you go past 6.8, you enter what's called acidosis really, really quickly. And if you go much farther past 6.8, you die. Same thing going in the other direction. If the pH of your blood goes too high, you go into alkalosis. And that's just as bad. And the reason for that is because all your enzymes and stuff start breaking down. You start decomposing and denaturing the proteins in your body. Now, what happens if we have an enzyme like this, and my reactants that are supposed to fit in this would be this shape, but instead, along comes Mr. Diamond here, and he fits in here, and he gets himself stuck. And can't get out. Because the open site, or the active site, tries to change shape, and maybe it like gets caught on our substrate. I don't know. If something like this happens, the enzyme no longer works. It would be deactivated. Now, this is just a cartoon, but some enzymes are susceptible to particular substances because they just bind irreversibly to them. Other enzymes can acclimate to a variety of shapes, and because that happens, molecules that are the wrong target molecule may come in, realize, oh, this is not where I need to be, and then lower the overall capacity of the enzyme to do its work. Kind of like, I don't know, going back to the factory example, if every other item on the conveyor belt for the assembly line was not used in the current production, you'd only go half as fast because you're only getting half as many parts as you need, right? So we can inhibit enzymes by just putting things in the active site that don't belong. And so this is called enzyme inhibition. Depending on what we're trying to do with the enzyme, inhibiting it may be a good thing, because for example, if you are producing things from an enzyme reaction, but making too many of them is toxic, then we may want to limit the production of that by inhibiting said enzyme. Now, if the reaction that we are trying to do with an enzyme gets plugged up by something that cannot be removed in any case at all, for instance, carbon monoxide inhibiting the iron active site of your hemoglobin, that would be very bad. That hemoglobin is deactivated and no longer can carry oxygen. This is why carbon monoxide poisoning is really awful. It's really, really difficult to undo carbon monoxide poisoning, because all of your active sites for all of your hemoglobin are just tied up with carbon monoxide, and you can't get oxygen to your body, and so you die. So something that like that would be an irreversible. I cannot speak today. An irreversible inhibitor is something that reacts with an active site and just stays there forever. It cannot be removed. In biology, that's very, very bad. You do not want your active site to be clogged up and unusable. Now, if you have something that just sits there and is like, oh, I'm in the wrong spot, don't mind me, I guess I'll be on my way, and then pops out and basically wastes time, that would be called a reversible inhibitor. Reversible inhibitors hop in the active site, realize, oops, this is not working out, and then leave. The overall activity of the enzyme is slowed down, but it's not zeroed. Now, why would a reversible inhibitor occur? Well, I have an example on this slide here. So the molecule succinate can be broken down into fumarate, and it has a specific shape as shown here. So I have two carbonyls with a CH2 in between them. So I'm going to put this into the active site. And I'm going to go hop, chop right there. I'm going to rearrange the molecule such that I make a double bond between these two, and then reattach. Now, in order for me to make a double bond, I would have to have two carbons here, right? So you can't make a double bond if there's only one carbon. So if we go down here to malonate, you can see that there's only one CH3 here. It's not a terribly different molecule. In fact, the ends look the same. So I've got this, it looks like a dog bone. Like, I didn't quite do the dog bone on the top one, but that's okay. So the molecule looks like the same shape, and it can fit into the active site. But when the active site tries to manipulate this molecule, it goes, hey, wait a second, I can't put a double bond here. I would break the octet rule. Because, you know, enzymes know what the octet rule is, right? And they say, well, there's supposed to be another carbon here. I can't work with this. This isn't helpful. And so this pops back out. And the end result is that cycle of the reaction is worthless. Nothing has happened. We have not produced the fumarate that we need for whatever facilitation we're trying to do. So this type of inhibitor is called a competitive inhibitor. Competitive inhibitors occupy the active site. Oh my goodness, what? My cat would like to announce that meow. I don't know if you guys have met Kirsten. You want to say hi? Say hi. Say hi. Hello. Say hi. He's shy. He's also very, very loud if he wants to talk, and I don't know what's going on. Now, so I forgot where we were. Competitive inhibitors inhabit the active site. So non-competitive inhibitors would activate or would inhibit other sites than the active site. So remember that if we have an enzyme that has a particular shape, if I were to bind something, say here, woo, now the active site changes shape, right? So if I do this, the active site is still free technically, but maybe it's not as ideal. In order for this other component to react with it, I would then have to push this out of the way, which might take a little more energy and slow down the reaction. So a non-competitive inhibitor slows down the reaction by just binding somewhere else in the enzyme. You can still use the active site. It just might require a little bit more effort. Using the cat as an example, who is just staring at me, you are a non-competitive inhibitor for the cat because the cat just wants to sit anywhere, and whether you are sitting or standing or lying down, the cat will still sit on you. It will just occupy a different spot. Maybe the cat is a non-competitive inhibitor for you. You can do what you want to, but the rate at which you do those things is slowed down because the cat on your lap wants you to pet him. Yeah, you want me to pet you, don't you? I pet you all day. Why do you have to... No, get away from that. Get away from that. Get away from that. See, the cat is slowing down class. He's a non-competitive inhibitor. I mean, I can get the class done. I can teach the lecture, but... This is all planned, by the way. So, when you think of non-competitive inhibitors, think of cats. Cats slow you down. They don't completely ruin your ability to do your job, but they do slow you down. Now, a subcategory of non-competitive inhibitors is what's known as feedback inhibition, and what this is when the product then binds to some other part of the enzyme. So if I had my enzyme here, so maybe I have this, and my product then binds over here to change the shape of the active site by doing that. So as I make more products, the overall reaction slows down because more and more products will bind to other parts of the enzyme. And as I run out of products, maybe like somewhere further down the line in the body has started using up the products I have, this inhibition gets reduced because there are fewer and fewer molecules to plug up the non-active sites. And so production would then increase. So those are all different ways that we can manipulate enzymes. Oh, I didn't realize that was the end of the chapter already. Those are all different ways that we can manipulate enzymes. So we can control them using temperature. We can control them using pH. We can control them by using the concentration of various substrate or enzymes. And then we can inhibit the reaction by using competitive inhibitors, non-competitive inhibitors, or irreversible inhibitors, which will basically destroy the enzyme. All right, that's your general overview of enzyme activity. So if you have any questions, let me know. Otherwise, we are pretty much done here. So we'll just put this on the screen. There aren't very many more of those. So let me get this on the screen here. So we're going to take our 10-minute break now because there's just not very much left to do. And hold on just a second here. Why is this not loading? There we go. Sorry, my timer was not loading for some reason. All right, so I'm going to start the timer. We're going to have the last 10-minute break, so we'll see you for that. And then we'll finish up nucleic acids, and we'll talk about what's going to happen. So, see you in a bit. All right, we are back. You'll notice that there's only one section for this unit, really. So I don't talk very much about nucleic acids because I realize that at this point, one of two things or both things will happen. So the first is we run out of time. Obviously, last day of week 10, probably not the best idea to cram in more stuff. You're not going to remember it. Like, who remembers what they learned on the last day, right? Well, hopefully you guys do. And then second of all, I don't know much more past this point. Like, as you can tell in the previous section with enzymes, like, I don't know what the hell acetyl-coenzyme A does. I'm not a biologist. I went and looked it up. It's used in the Krebs cycle. Like, I would not have known that. I would just be guessing. I was trying to guess by looking at the molecule, but that's not super effective when the molecules are so big. So my ability to do chemistry with these molecules kind of breaks down at this point because they are so complex, and I have spent all of my time studying simpler molecules. So the point is, at this point, we are essentially reaching the limit of my knowledge. So if you go past this, you know more about me in this topic for sure. But I can tell you what I do know, and I think that DNA is a simple enough molecule in terms of, like, its components, at least, that we can talk about that for 5 minutes. Like, sure. I'm not going to tell you how to transcript DNA and how to use mRNA to code stuff, and we're not going to talk about uracil. I'm going to mention uracil, but we're not going to talk about where that comes in. I just care about the molecules, so that's what we're going to talk about here. DNA is made of 3 parts. The first part is nucleotides. So there are 4 nucleotides. We call these base pairs. And then there's this 5th one that is used in RNA. So the base pairs are adenine, thymine, guanine, and cytosine. Now I'm going to highlight them in the order in which they pair. So adenine, thymine always pair together, and then cytosine and guanine always pair together. And the reason that these pair together is because they hydrogen bond to each other, and their shapes just particularly complement each other well. Now, why are they called base pairs? Well, it turns out that they're basic because they contain nitrogen. So that's why. Again, we're not going to talk about uracil. It's used for RNA synthesis. I want to say there's a picture of the... Yeah, here we go. So adenine and thymine pair up with each other, and guanine and cytosine pair up with each other too. I wish I had rotated these in the correct orientation because they both hydrogen bond with each other effectively, but I don't know which side of the molecule is the most effective for that. So what I'm going to do is I'm going to go look that up real quick. So hydrogen bonding. Let me see if my guess was correct. Wow, that was totally not where I would have guessed for adenine and thymine, but guanine and cytosine worked out pretty good. Let me just copy these pictures real quick. So copy. That's not what I wanted. So we'll paste this one. Oh, Google, why? All right, here's what I'll do. I kind of wish I had this picture in here, so... So it should just pop up right here when I do this one. So here's adenine and thymine and how they hydrogen bond together. So you can see that the NH on thymine is kind of the only opportunity it has to donate a hydrogen bond. And it's going to point that at one of the nitrogens on adenine. And then the NH2 group on adenine points back towards the O to make a pair of 2 hydrogen bonds. And then I should be able to do this. And there should be guanine and cytosine. Yeah, there you go. This one's kind of a duh. This is the only orientation that gets you 3 of them. So, making multiple hydrogen bonds means that those molecules will stick together very effectively. And so their shapes and the positions of all the nitrogens and the NH groups and the oxygens just happen to be such that you can get this effective hydrogen bonding. And so these base pairs will pair up in these orientations every time because they've optimized that hydrogen bonding. Now, what do we do with these 4 things? Well, we're going to attach them to the second component and the third component, which are deoxyribose and then phosphate. So DNA consists of the base pairs, which go in the middle, but the backbones along the sides consist of 2 components. The first is phosphate, and then second is this sugar, deoxyribose. And we just alternate these over and over and over and over and over. So the way that this works is this OH group here is going to make a phosphate ester. And this OH group here will just go to the next phosphate ester. Where does this OH group go to? This is the glycosidic linkage that goes to our base pair. Now, knowing those bonds is good, but I'm not going to make you draw this structure on the exam or anything like that. So what we're going to do is we're going to reduce this down from this structure to this structure. Here we go. So we'll go back to this because this is just, I find this the more interesting structure. So here's our phosphate ester. Oops, not that one, sorry. This is also phosphate ester. And then this connection right here is the glycosidic linkage. The glycosidic linkage. I don't know where that G came from. And so we make a polymer. The backbone of DNA just repeats over and over and over and over and over and over and over again. So here's our structure for DNA, just simplified to an extreme. So we've got phosphate, sugar, over and over and over and over again. And every time we have a sugar, we can connect to one of the bases randomly. So the base could be A or T or G or C. But then the base across from it will always be the complement. So if I put A here, I have to pair it with T. If I put G here, I have to pair it with C. If I put C, I have to pair it with G. There's no requirement as to which side of the double bond they appear on, or not double bond, of the hydrogen bond that they appear on, because it's essentially random and both sides are essentially the same. Now, when you do this enough, you create a strand, and the nature of the deoxyribose and the phosphates is that they can be coiled into this spiraling structure, which is the double helix of DNA. I had to tilt the picture sideways so that it would fit. But yeah, that's DNA. So if I was going to ask you this question, draw a simplified structure of DNA, if one strand contains the bases TGCA in that order, this is what I would have you do. So we would say, okay, so I'm going to draw the backbone here. And so every other one would have a base on it. So this would be T. This would be G, C, A. And then what are these connected to? Well, bases are connected to deoxyribose, which I'm just going to call the sugar. So sugar, sugar, sugar. And then phosphates go between these, which I'm just going to call, I'm going to do that. And then I would just mirror this on the other side. So what pairs with T? A. What pairs with G? C. What pairs with C? It's G, and what pairs with A? It's T. And then I would do the same thing. So this would be the sugar. Really, the main issue with doing this type of question is just drawing the damn thing over and over and over. So this would be phosphate, 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 sugar, sugar, sugar. There you go. So that would be the simplified block diagram for DNA. And it turns out that's all I want you to know for DNA, is I want you to know which base pairs pair together. So T pairs with A, G pairs with C, and then the generic structure of DNA. So like, what are the components? So there's the base pair, either A, G, T, or C. That connects to a deoxyribose, which is a sugar, through a glycosidic linkage. And then the sugar itself is phosphate estered to phosphate groups. All right, if you have any questions, let me know. That's really all I have. So yeah, so there you go. All right, the last thing you have to do for this chapter, and for any chapter really, is to celebrate. Hooray! The end. That's it. That's all I got. This is the part where you celebrate. Yeah, that's all I have. So I'm going to go to this screen really quickly, which is going to be covered by a black box because, well, you know what, we just don't need this anymore, so this can go away. And let's talk about what's going to happen for the next week or so. So today is Wednesday, week 10. Unless you are watching this video right before the exam to study, in which case, hello from the past. Is everything okay in the future? You look well. But tonight, I will post the quiz for chapters 18 and 19. I don't remember what number that is. It's 7, maybe? 8? I don't know. And what day is Friday? Friday is the 12th. So this quiz you only have 2 days to do this quiz. Well, tonight also. So you'll have tonight, tomorrow, and then Friday. And it will close Saturday the 13th. I almost wrote 23:59. I'm used to military time, sorry. I'm not in the military, that's what we use in the lab was military time. So after the quiz closes, the answers will be available from Sunday the 14th. So you'll have the answer key available all day Sunday. Presumably you won't really need it because the last unit is pretty short anyway. And then finals week, which is week 11. Monday is exam 2, which covers chapters, oh gosh. I had to look that up. I think it's 15 through 19. Yeah, so wherever 15 we got kind of stuck in the middle of during the first exam. So whatever the second half of 15 is, wherever we stopped, all the way to the end. So all of the biochem stuff is going to be on the same exam, but there is going to be a little bit extra organic. So like esters and carboxylic acids and amides and amines, that's all going to be on this exam too. So you can't forget your OChem. So let me write that down here. So that would be chapters 15, so the end of 15, to 19. And then when is my final exam scheduled for this class? So I have to look that up. I usually put stuff on Thursday, but maybe I put this one on Wednesday. I put it on Wednesday because this is a Monday, Tuesday, Wednesday class. Okay. So the syllabus says that we would have a review session and Q&A on both Monday and Tuesday, but because I'm moving the exam, we're going to have the review session on Tuesday. So Tuesday from 10 to 12. So what would normally be class time will be the review session. And so I'm going to ask you, please, please, please bring questions to the review session. Otherwise, I'm not going to know what to review with you. Okay? You can ask me literally anything about the class, except for from the exams, because those will be locked. But anything else is fair game. So any quiz question, any worksheet question, anything like that, that you have a question about that you want me to answer so that you know it for the final, please bring it to that review session. Okay, so we'll have that from 10 to 12 on Tuesday. And then what else will we do? Oh yeah, obviously the final. I'm going to do this. Because honestly, at this point, I don't care when you take the final. So I'm going to open the final for 2 days. It will open on Wednesday. It will close at the end of Thursday. And at some point in there, take the final exam. You get 2 hours to do it, and you do still get 2 attempts. So take it once on Wednesday, take it once on Thursday if you like. Or take it once, do really well, and then don't take it a second time if you feel that you did great. And then there is one more thing that I do want to post. So lab number 5 and last call for all NQA. And that would be Thursday, and I don't know what number day that is. That's 18? Yeah, 18. Okay. So this is actually really important. Let me make sure that my syllabus... Okay. So the syllabus says lab worksheet number 5 is due on Tuesday. If you need an extra day or 2 to do that, that's totally fine because I'm not going to grade everything until I get it all. So I will leave I will have the due date be the 15th, but the last call deadline will actually be the 17th. I totally messed up those numbers, didn't I? Yeah, I totally did. I'm sorry. So on the syllabus, it says lab number 5's worksheet is due on the 16th. I will leave it open on the 17th and also on the 18th. But every assignment that you want me to grade has to be turned into me in my mailbox by the 18th. I cannot accept anything on the 19th or later because starting on the 19th, I basically turn off the internet and I just grade until everything is done. I don't talk to you guys. I don't answer email. I do everything that I need to get all my grading done because grades end up being due the following week. And if I don't get grades done by then, and I have a lot of grades to do, the registrar gets really, really, really upset because he has to deal with putting all of the grades into the transcriptor and doing stuff like that. And if I screw up his assembly line, I do not want to be the person that messes that up. Also, Carl is super, super awesome, and I don't want to make his day any worse than it is. Our registrar is fantastic. So yeah, that's everything here. I'm trying to zoom this out. There we go. There you go. So that's the last bit of information that I have for you. So that is the final agenda for our class. Make sure you copy that or hit pause. Like, you can pause the video, right? And that's it. I want to thank everybody for a wonderful quarter. Thanks for coming out to the lecture. I see that there are still 5 or 6 of you here. So I know a lot of you came out for a lot of the lectures, and some of you came to every lecture. And I realize that that takes a lot of time during a quarter where it's all online. But whether I got to know you in the chat, or whether you send me questions over Discord or email, or whether you never interacted with me at all, and I have no idea who you are, thank you. Thank you for making this a memorable quarter. And thanks for bearing with me in my first time teaching Chem 131 in, God, who knows now how long. So that's it. Thanks for everything. Thanks for watching. And I will just leave you with a last little tune to say goodbye. Send me any questions you have over email. And have a good night.